I will actually talk about what is going on in Lausanne, in Switzerland, at the University Hospital, where I opened the lab in March 2021. So it's brand new, the laboratory of bacteriophages and phage therapy. And I will talk about really the manufacturing part of the project we are trying to develop, which is called PhageEU. And this is registered in the strategic plan of the University Hospital in order to bring phage therapy to patients. So I will focus on production, and here are the objectives.
We had problem with your connection.
You don't hear me?
We'll switch off cameras, all of us, and maybe then the connection will be better.
Okay, so I hope it's better. Do you hear me better now?
Yes, yes. Let's try like this. Yes,
okay.
Go ahead.
Hopefully it will work. Otherwise, I am very sorry.
It's fine.
I'm at home right now, so I don't know how the connection will go. So as I said, I will focus on production, and the objectives are listed on this slide. So we want to establish a phage therapy production pipeline based on our institutional phage collection. We have more than 300 different phages right now in the collection against the escape. Not all the phages were isolated by my lab. We have also phages from many collaborators like Benjamin Chang in Yale. We have also phages from the Christine Bourcel in France. We have other collection. And we want to actually produce our phages in GMP, because we need a Swissmedic approval, because one goal is to treat patients in frame of a clinical trial that we are trying to develop right now, but also in the compassionate use framework, we would like to have authorization from Swissmedic or the cantonal pharmacist. So the goal is, as I said, to bring phage therapy to treat to patients in a personalized framework. Okay, so the objectives. There is one point in the law in Switzerland which allows you to produce small batches of medicines in an academic setup, but still here the requirements that are needed to administer these phages to patients is that you need to have a GMP license for phage manufacture, because phages are classified as biological medicine also in Switzerland. So this is the PhageEU personalized treatment pipeline. So as soon as we have a patient who has troubles with antibiotics, he is not responding anymore, we get the strain in my lab, in the phage lab, and there we are doing phagograms. And this allows us to select 1 or 2 phages maximum that we use to treat patients. And we propagate these phages in 1 liter batches, in order to obtain what we call a CBP, which stands for concentrate of bacteriophages. Okay, are you still hearing me well? Just to check.
Yes, we can hear you well.
Okay, great. So this CBP, before being delivered to the CPC, the Center for Cell Production, which is in GMP certified, you will see that later, we dilute it in my lab. And we are doing the propagation and the dilution of this CBP in the GMP. It's even more than principle, because the only thing that differs from the CPC is that in my lab, only the room is not classified, but all the rest is full GMP. Okay, so then at the CPC, they receive this diluted CBP, which is the starting biological material, and they purify the phages from this CBP in order to get what we call the SBP for the suspension of bacteriophage, which is the final product. So we will go through this pipeline of production very slowly. So in my lab here, you can see the room in which the technicians are preparing this CBP. We are starting from bacterial working seeds and phage working seed. Okay, so let's say we identify a phage that is very active on many different strains of the patient. We are producing it as a concentrate of bacteriophage, okay? Because this product is also very stable in saline at 4 degrees. So it's an intermediary product, you will see. So we start with this 2 seeds. We mix them in 1 liter of LB animal-free. We are focusing on Pseudomonas aeruginosa phages right now, but we could adapt the pipeline for many different phages. After an incubation, we get this native bacterial lysates, and then it's a series of centrifugation and frontal filtration that we use in order to remove bacterial debris to prepare a bacterial lysate, which is devoid of bacteria, okay? And then we have other centrifugation in which we pellet the phages and we resuspend them in simply saline in order to get a CBP which is concentrated at 10 to the 10 to 10 to the 14 PFU per ml, and this is stored at 4 ± 2 degrees. And it's very stable, as I say. We are doing stability tests, and here you see we have produced 5 different phages, 4000 to 4004, and you see the stability here, which is in PFU per ml over days. And for instance, if we take this one, we have 6-month stabilities, so it stays at 10 to the 12 for, I hope, forever. I presented this in Australia. Now you can add 3 months more, so it's 9 months, and the stability is still the same. Okay, so these products are very stable, but they are very dirty also. When a new patient arrives and we get a new strain like this one, PAE0560, then we test these 5 CBPs using turbidity assay. So this is our phagogram that we are performing through turbidity assay. We test an MOI of 1, and here are the results. So OD in function of time over 24 hours, the bacterium without phages, and then different phages that are added. And you see that then when I report to the clinician the results of this phagogram, this is the way I am doing it. So 4000 in green would be high for intermediate susceptibility because there is regrowth of resistant strains. Here also for 4002, and then the 3 others, we score them S, okay, for susceptible. If you would have a curve like without phages, it would be R for fully resistant. Then let's say we decide to produce 4001, then we go back to the CBP, we dilate it simply to have a concentration between 2 to 10 to the 8 to 2 to 10 to the 9. This is due to the endotoxin that we need to remove in the final product. And this we deliver to the Center for Cell Production, which is in full GMP. It is certified for ATMPs, and you will see later that we have now the certification for phage production in GMP also. They start from this diluted CBP, and they are actually producing the SBP, which is 48 flasks of 1 ml concentrated between 10 to the 8 to 10 to the 9 PFU per ml, in which you have the endotoxin below 175 EU per ml, because we treated a patient who weighs 35 kilograms. So because it's 5 EU per ml and per kilogram of body weight, we had to be below this number to be able to inject the phage IV. So in this process, you have again filtration, and then we are using simply the pierce resin in batch mode to remove the endotoxin. And if we are below this number, because we dilute twice here, then it's okay, we can dilute and filtrate and get the SBP, the final product. If we are above this threshold, then we do a second purification before dilution and filtration, okay? So this is very interesting because we discovered something very recently. You see in black here, these are the histograms, the stability for the SBP 4000, also 1, 2, 3, and we didn't do it for 4004 because we discovered a non-disclosed, I will not disclose what we add to this SBP to actually stabilize them. You see that for some phages like this one, we had a stability for less than 8 days, because after 8 days, you see we had a titer of 10 to the 5 instead of 10 to the 7, which was the limit that we fixed ourselves for the stability. But when we add this stabilizer, you see now that we have also here, at least 6 months of stability, even in these SBPs, which are diluted and also in saline and cleaner than CBPs, huh? Okay. So that's a very good news. Here I show you the quality controls that we are doing on different things. So we are performing identification by full genome sequencing of the bacterial working seed, the phage working seeds, of course, in the CBP and in the SBP to be sure that we have the right phage that has been produced, and also no other phages, no temperate or no contamination with another lytic phage. It happens sometime. Huh? We check, of course, the activity on the patient strain using phagogram at different steps, the bioburden with the BACTEC technology on different products. Phage titer, we use diluted drop test to titer the concentration in the highly concentrated products. And then we switch to double layer assay to be more accurate because the titer is really lower in the diluted CBP and the SBP, the final product. We check the endotoxin in the SBP. We have also a threshold here from the sequencing. We get also the reads that match to contaminant bacterial DNA. And we did put the threshold at less than 0.1% of all the sequencing read should match to bacterial DNA. So that's the specification. We do that also in the SBP. Total proteins, we do that as an R&D project for the moment. We do not have any specification. And we sequence all the protein in the final product. And there we see that we have very low amount nanograms or picograms of contaminant proteins in these SBPs. So it's quite encouraging. And we will see if we will add specification sometime for these total proteins that are present in the final product. Batch in August 2022, and you see here the first batch that has been produced for the file that we submitted to Swissmedic. So here is the director of the Center for Production. This is the chief of the pulmonology at CHUV. This is the infection disease specialist, Benoît Grégory, and the other people on the slide. And here is, finally, I'm very happy to tell you that we are the first academic institution worldwide, I think, to have this authorization, this certificate for GMP production of phages. And this has been granted last year in June, as you see. So that's a very nice achievement. Of course, plenary program, you have many services at the hospital that are involved, like also the pharmacy, of course, and the Center for Research and Innovation of Clinical Pharmaceutical Sciences, so in which my lab is located, actually. This is the team with many physicians, my boss, the people at the CPC, and also we have jurists for question of legal affairs when we talk with Swissmedic. Many people, the technician in my lab are involved. Also other people at the CPC, the pharmacy, the Institute of Microbiology, who provides us with the strains and are also working on the pipeline for the phage genome sequencing, the proteomic facility. And I thank you also for your attention.